So Harshad is very dynamic. Okay, he does several things. Uh, uh, Even like being his PI, like I couldn't handle so many things the way he functions. Okay, all the time hyperactive. Okay, because he's managing 1,000 things at a time, which I couldn't. Okay, so Harshad did his PhD on butterflies. Okay, the pupil coloration from Trivandrum, uh, ISO Trivandrum, one of my collaborator Ulasa. He's an ecologist. So he did wonderful work. He was the guy who discovered that when pupae are of two colors, brown and some green colored, and butterflies picks the places, okay? So the larvae settles down somewhere based on the humidity pattern. If it's drier, those pupae will end up something else. And if the wet habitat, those pupae will end up something else. They did a wonderful work. So proud of him during his PhD. So then he came to our lab and then he's continuing that work, but in a different system, like flies, which are way, way smaller. So he had lots of trouble, butterflies too big and the fruit flies like hardly 2 millimeters. So he adjusted his lenses and all that. So, but he's doing wonderful work. He has selected like 6 different genetic lines, and now he's playing with the pigmentation patterns. Like when you start from in a different genetic background, and if you're selecting for a common stressor, how does that end up these patterns? And so it goes all the way from phenotypes to the genes and all that. So, but today I don't know what he is going to talk, but he always tells wonderful things. So I just stop here. Okay, Harshad.
So thank you, Subhash, and good afternoon. Good morning. And I'll not talk much so that we break in time for our tea. I should first thank Subhash, in whose lab I joined as a postdoc in 22, and all the beautiful lab members. And actually, science is more fun when you have exciting people around you. If you want to do something, but then you don't have support, you don't have like-minded people, things don't happen. So I was very fortunate to join Subhash's lab because he has given me absolute freedom to explore things. And obviously, there are a lot of interesting things already present in the lab. So I didn't have to scratch my brain when I came here for postdoc. There were like 1,000 ideas lying, and then Subhash said, you're free to pick them. So apart from that, I have wonderful colleagues. We have Abhishek and Aviroop here. There are others, Divita, she's not here. And there are a lot of people who come in the lab and they add to the spice of the lab. And with this, I would now shift our focus to what we have come here for. So I hope you guys are all enjoying this small workshop. It's a very personal one. We have a lot of interpersonal communication. So I think that is the best memory we can have from any workshop or conference. And to begin with all this hue and cry about climate change and population declines, biodiversity losses, I think that is what is the some of the few hot listed words in today's news. All of us want to talk about it. All of us want to be in news because of these things. We want this almost climate change and biodiversity loss has become a page 3 word nowadays. So we are all concerned that, of course, if biodiversity is lost, then we'll not have a healthy planet. And I think a lot of these things were already covered yesterday by Charlie and a lot of taxonomists. Dr. Ramamurthy also spoke about it briefly. And then we also had a nice talk by Priyaranjan now. So we think that the earth is warming. And of course, it is warming. If you see, it's red and yellow. And a very little portion is fainter or paler blue. But actually, the earth has been like that for quite some time. I mean, the earth began as a solid ball of fire and then it cooled and then it expanded. It was hot again. It went through a lot of hot and cool cycles. So then why are we, making such a fuss about it? So right now, Annie just pointed like, why the hell do we want conservation? I mean, let things be. And Prayer Engine was saying, yes, let them be. Let people eat fish. Do not protect the estuary. Let them have their natural lifestyle. Do not go and protect anything as such for protection's sake. And I kind of partially agree with that because if we actually had this geological history where the earth was cooling and getting hotter and still animals were getting produced, you had a lot of biodiversity generated, why are we worried about climate change, which is just a recent phenomena, like perhaps 50 to 70 years ago? And turns out that Yes, climate change does have an effect, because I think one of the things which you might feel bad is I'm going to be anti-anthropogenic. I don't like what human beings do, because human beings have conveniently said it's climate change, and climate change caused a lot of these mega herbivores to go extinct. But they are not saying what they actually did. So mega herbivores went extinct, they came about again, and they still blossomed, they flourished. But when human beings started invading all continents all over the globe, I think that is where a lot of biodiversity losses started significantly. So there's a constant debate between ecologists and paleontologists as to where the origin of these extinctions began and what was the pattern. Was it anthropogenic or was it climate change? So some say because it was suddenly cold, they couldn't survive the freeze. Some say it was suddenly hot. So actually, many people also are of the opinion that many of the mega herbivores went extinct because of human interference, because humans could kill, humans had developed weapons by then, and that is why you lost a lot of herbivores. So you can see in this picture that the size of these herbivores or any animals present on the earth way back was quite bigger. I mean, it's as tall as the tree. And then you can see over here, the dimensions have gone so low. So maybe we have lost a lot of species, and whatever species exist, we have tried to belittle them, literally, in the literal sense. So you can see that amongst the mammals which are over here, the ones which are smaller have managed to survive because they bow down before us. We can control them easily. But the ones which were mammoth in size, we have managed to finish them off, so that our egos are satisfied. And you can see that out of 48 mega herbivores, which were of the woolly mammoth size, almost 40 have gone extinct. But if you see some smaller mammals like langurs or some of these dogs or animals which are relatively faster, but they are still of an intermediate size, they have still survived. So maybe we are kind of discounting the anthropogenic effects which we have done on this planet, and we are just blaming it on climate change all the time. But climate was changing. So what changed is our behavior, perhaps our greed. So that's a contentious topic. Now coming back to insects, and then we think insects are on the decline. Come on, obviously if a mammoth can go extinct, then these tiny organisms, I think we don't even know how many exist. So we were just discussing about this, that what is the insect number? And Wilson said, I don't have any idea. We don't have even a slightest magnitude about what is the diversity of insects present on this earth. But definitely we know one thing as human beings, that insects are important for us. So we get fruits, we get vegetables, we get all lot of things. Insects are like the best ecosystem servers. So we can manage without tigers, as Priyaranjan said rightly. We can manage without elephants, but I think we cannot manage without bees and butterflies. So of course, it is important to know whether they are really going extinct. Now, I always see it from the other side. So everybody says that it is half empty, all the insects are declining. But I see that there are some insects surviving. And why do we not once focus on this side, that irrespective of climate change, irrespective of global warming, there are still a lot of species which are still surviving? And have we ever thought a moment why they are surviving? I mean, of course, there are species which are dwindling down, which are not able to cope up with environmental change. But then there is certainly some amount of species which can tolerate that change. So if you go and check on this website, which is the International Union for Conservation of Nature, they have a lot of data about how many species are actually living today, how many have gone extinct, and how many are still alive, extant. And with respect to insects, what is this number, 80%? 80%, do we know 80% of the insects on the earth, or we do not know? Sorry, we don't know, correct? So if you have a full circle, out of that yellow, the 80% is the part which we don't know. So this website just reports some 1 crore or 1 million insects, and that is not their true number. Now, if they say that we have discovered just 80% of the extant species, the ones existing now, you can imagine we do not know 80%. That means we just know only this 20%. And that means you can get the depth of magnitude of what is the sheer diversity of insects. And if we don't know them, then do we really talk about decimating insect numbers? I mean, we are saying insects are declining. You don't even know 100 species. And then you are saying 90 species are lost. You don't know if 90 are lost, there are 90,000 out there. So definitely we are not losing out on some. But definitely it needs addressing. So the limited knowledge which we have got about insect declines is coming from a few studies around 3 eras of extinction. So we do not exactly know how many mass extinctions were there. We assume there were 5, but in case of insects, there were just 3 for which we actually have records. And if you go to see a mammalian record, you will have a lot of evidence, but we do not have many evidence of insect fossils. And all of these are actual fossils which are discovered of insects in these past extinction events. And you can see there are far and few. So these do not actually explain the extent of insect diversity even back then. And we have very little idea of what is the current scenario. So you have a few insects which are preserved in amber, you have a few flies which are there etched in the stone. But surprisingly, you have very less evidence for terrestrial fossils, for insects which were terrestrial, compared to what existed in the waters. So all of this insect decline talk is based on these insects which are water living. And because we have evidence, because their shells can easily be calcified, and so you have lots of fossils of these. So then when you have most of the data or evidence coming from aquatic organisms, how can you say that insects on the land are actually declining? So we should think about these things whenever a research comes out and people are just talking high that something is declining. We do not know what is the reason, what data is they are actually considering. So most of the fossil data of insects come from these water bugs and aquatic organisms. So definitely we have less evidence to say terrestrial insects are declining. Some other sources of knowing insect numbers or diversity is going to museums and looking at all these beautiful specimens of butterflies and coleoptera beetles preserved, and that's it. We are happy with watching charismatic species. We just like beautiful things. And if I present that dung beetle which he showed, which was so beautiful, it was iridescent, but there are dung beetles which will be plain obscure brown. There are butterflies which I was working on, they were brown, and then when people used to say, you work on brown butterflies, brown is not a color. So there are unpleasant things, but they do exist. They do help in pollination. And now if you think you are just going to limit your knowledge of insect diversity to these few specimens, we really do not have any idea about the insect diversity on this planet. And another is this business which everybody of us does at some point of their life in ecology and evolutionary projects. We have to go to the fields, we do some monitoring, and we go for a field visit maybe once in a year when the weather over there is cool. So we go to Jammu Kashmir in the winter. It's a very nice weather. You have a lot of exhibitions and you get good food and tourists. And I think that is when we do the sampling. And then how biased we are in our sampling. So I collect some insects in the months of December and January, and I don't go there for the rest of the year. And precisely when I don't go, there are a lot of insects over there. So we are missing out. So what I want to say is when we do surveys, biomonitoring, we do tend to have a lot of biases. We try to go to places which are quite accessible to us. So I'll go to a place which is just near my house, which has some trees, and I call that a mini forest, and I collect data from that, and I say, yes, I found some 50 insects. I have actually not gone to that older, wilder areas which have a lot of diversity. So there are obviously a lot of caveats in our methods of assessing insect diversity, which is why I say that we really do not have a true taste of it. Now, evolutionarily, if you see whether insect extinctions were a real thing, you would actually see that over these epochs, insect diversity, in fact, has gone increasing. And if you consider insect extinction, extinction is when species cannot be recovered. Origination is when you have new species form. So when origination is exceeding extinction, you basically have a high turnover of insect species, and that is what we call as diversification. So in case of the class Insecta, one special feature is they are actually diversing all through these years. Their numbers have not gone down, or nor have they been at a similar pace. So if we consider this diversifying trend in the line of declines or extinctions, it's not really true, because if you see, it's an increasing curve. So maybe there are some insects which are going extinct, but then there are 10 others which are coming in their place. And if 10 others are evolving in the face of climate change or high temperatures, then definitely there is something adaptive about these insects. That means they can actually evolve. They have the evolutionary potential, or they have the capacity to adapt to these changes, and they can still form new species. So when we actually talk about declines, we are actually making a very turning a deaf ear or an eye to these numbers where the diversification rates are high. So now let us see what is actually diversifying. So if you just consider this tree in which you have your insects at the very base, maybe the wingless insects, and there was a common ancestor, and then you have your odonata, that is your dragonflies, you have your cockroaches, you have your katydids and stick insects on one side, and then you have your Diptera, Coleoptera, Hymenoptera, Lepidoptera on these sides. And now if you see over here, the insects are quite smaller. And what I would like to point out is what criteria do we actually have apart from the morphological features? And turns out that you have a lot of genomic information which could help us. So I think that is why I would thank Saroja, because she has laid a very perfect foundation for your understanding, and I can rely on that and skip over a few things. So if you just look at genomes, and you have a careful look at these genomes, you can amass a lot of wealth about insect diversification from them. So if you see this particular branch, the right branch in general, you can see that these insects are the ones which have in general a higher diversification rate. Why? Because initially both of these branches had the same genome size, but over years they have evolved to have larger genome size. So all of these stick insects and cockroaches have large genomes, around 10 times than this. But what is the characteristic of this is their genomes reduced, and now in recent years, they are actually increasing. So I'll show you in a few minutes that when you have a reduction in the genome size initially, afterwards it's increasing, and that increase in genomic size could be adaptive. So these are these four distinct orders: Lepidoptera, Hymenoptera, Coleoptera, Diptera, and Lepidoptera, which are having the highest diversification. And you know that beetles are, I think, 1/3 of the world is beetles. Like in plants, they say 1/3 of the world is orchids, because orchids are so speciose. Similarly, you have speciose beetles out there. So why we say beetles are speciose? Because they have been diversifying all through this time when we thought that insects would actually decline. And we have no good idea about how good they are at multiplying in the number of species. So if you actually do a comparison of genome size, so on your x-axis, you have your genome size, and these are just the different taxa of insects. You can see that the taxa which are in red are actually those four. They have smaller genome sizes compared to the others, but these are the ones which are diversifying. So what is hidden in the smaller genomes that the bigger genomes do not have? And one thing over here is all of these classes with larger genome size have actually not reduced in their species diversity. So if there were 50 species of Odonata back in 50 million years ago, we still have around 15 of those. But you have a lot of turnover in these species, which means that you have a origination or new diversification happening. You have new species being produced if older species are going extinct. So we thought that we should use this genome size as a rough estimate of what could be the basic evolutionary potential of insects. And turns out that in general, if you see, so this is a paper we published recently, a review paper, and me and Subhash have worked on it. And if you see over here, you have some traits. And some of these traits are physiological, and some of them are related to your fitness, which means your reproductive potential. And if you see, these are the coefficients, correlation coefficients, and the direction shows that it's a positive or a negative correlation. And C value is basically the haploid value of the genome. And these are the species. So if you carefully look, this is in general Drosophilidae. For some 10 to 12 species, you have higher genome size. And if you have a larger genome, you have a larger body, you have a faster male maturity post-eclosion, you have a higher X size, I mean a bigger X size, your blastoderm is more thicker. In case of coleopterans or beetles, you have a higher rate of fertility. You can compete a lot if you have a higher genome size. Your mating success increases if you are a male, and the size of their genomes, you can say, is 0.71 to 1.42. So that's quite large compared to even these Drosophilids. And if you then see another class of Coleoptera, their X size and fecundity increases. So obviously, these studies are from different species, but all of these studies point out to the fact that in general, if you have a larger genome, you could have a bigger adaptive potential as well. So we actually need to test what is it. I mean, when we say adaptive potential, it's very easy to say, but unless we measure it, we will not be able to say. But yes, this is an indication that some of the parameters might get affected. The second thing we are talking about is transposable elements. So when you say genome size, what is your idea of genome? A genome has a lot of elements which are not coding. Some of them are coding. A few percent of our genome is actually coding. But then you have these transposable elements. I think all of you are aware of transposable elements. Who was the discoverer of transposable elements? Barbara McClintock, correct. What was the system she discovered them? I mean, where did she find transposable elements? In maize. Okay, great. So she found that in maize, there are these kernel sizes which are getting varied, and you have a lot of variation in respect of the color. And that is because there were genes which we call as jumping genes. They just skip through the genome, and the accumulation of these genes, the repetitive sequence of these genes, is what we call as these transposable elements, because they can move freely. They are often called selfish. We do not know what is the purpose, but we have found out that there is a correlation that the higher the proportion of transposable elements in your genome, the larger is your genome, and the larger is your genome, the larger might be your adaptive potential to environmental stress. So I'll just particularly point out to this Aedes albopictus, because you can see that the percent repeat, the percent of transposable elements which are repeated for Aedes albopictus is 51% of its genome. So almost 50% of its genome is repeatable with transposable elements. So we do not know what could be, but turns out that Aedes albopictus is an invasive species. It can adapt to different habitats. Its eggs can sustain diapause for a long period of time. Its larvae can sustain a lot of filthy waters for a long period of time. The adult has a capacity of staying dormant for a long period of time. So we do not know if all of these properties are attributable to this larger genome size because of your higher transposable element content. So some of these things need attention, and as you can see that there is adaptive significance attached to some of these. So some of the larger genomes like grasshoppers, we really do not know. They have a big genome size, so 70% of actually the genome is repetitive, but there is no significance. And that thing I'm telling, that these species have never had any high diversification rate. So these grasshoppers have been having these transposable elements for a long time, and there has been no diversification. But in the mosquitoes, you had a reduction in the genome size in dipterans in general, and now your transposable elements are increasing. So we do not know if these transposable elements are adaptive, they could be retained. If they are, deleterious, it will be purged out from the genome immediately. But there are other advantages. So in Biston betullaria, you have 5% of TE, which is repetitive. So what is this reference of Biston betullaria? Does anybody remember? When did we study Biston betullaria in our coursework? Perfect. So do you know why were you getting a carbon moth in that time? Why were you getting the dark phenotype of moth in that particular time? Yeah, but what was it coming because of? Why do you get a black color? Fat is more. Yes, but factories is an external cause. What is the change in the moth which is causing it to become a dark form or a light form? Yeah, but adaptation through what? I mean, did it go and get a cloth of black color and started wearing a black sweater, huh? Predation. From predation, okay. But what was the predation selecting for? Yes? It's fine, it can be wrong, you can just say it out loud. Okay, so when you have this variation, when you have anything happening in evolutionary biology, you first need to have a raw variation present in your genome. So you should have alleles which will code for a darker phenotype. You should have alleles which will code for a lighter phenotype. Now, as I said, because you are having higher predation, some of the alleles which were producing the black color got selected for because black beetles could survive, or black moths could survive, and that is why the black alleles increased in frequency. But the actual reason behind this is in those black alleles, you have a transposable element sitting over there, and that transposable element is sitting in the optics gene. Now, optics gene is the one which is related to pigmentation. So if optic gene has a transposable element, it gets switched on, and then you have the production of pigmentation. So when we say that there is predation is a selection force. Now, selection doesn't act on the organism per se, it acts through genes. So when you have something special in the genes, that is what is going to get selected for. So what actually was getting selected is that allele, but in that allele, you have a transposable element. Now, had this transposable element not been there, you would have had no selection. So when we talk about industrial melanism, it's all because of transposable elements. So now if you can think that so much is the complexity of transposable elements, we have not actually detected what is their adaptive significance. So much of it remains to be done. And that is where we would like to take the help of genomics. And I would say that, okay, what is the application of knowing so much of transposable elements? Why do we need? So we are talking about insect declines, and definitely insects are declining. But we need to find out pockets where insects are actually going to decline. So if you want to now apply this strategy of your genomics into conservation, where do you go? Where do you find out? How do you find out where insects are declining? Any guess? So if I ask some of you that go to some place where insects are declining and give me those insects, I'll start sequencing them. Where will you go? Any idea? Cities. Cities insects are declining. Okay, where else? Cities, there are no insects, in fact, I guess. But yes, there are. But okay, where else? And in cities, let me tell you, the environment is quite variable. You have seasons which are not clear, but you have a lot of variations in the temperature, humidity because of your fog or your smoke and a lot of other factors. But one thing which we should keep in mind is you should actually go to habitats which are stable, because these insects have never experienced any changes in their climatic variability. So if you go to the rainforest of Amazon, or even in India, if you go to the rainforest in Kerala, in a few places or northeast where environmental change is very limited. So you have rains throughout the year, you have fog throughout the year, you have a lot of precipitation throughout the year. So those genomes are somewhat not going to adjust to your rapid climatic changes. So now in these regions, if the sun shines brighter, if the temperature increases, these organisms which have stayed in this habitat for long, they do not have any adaptivity in their genomes. So this is just a thought. So now if you go and sequence these genomes, you will find that these genomes have a lot of, I mean, their genome size is not that high, perhaps. And if their genome size is high, maybe they are also experiencing a population bottleneck. So the gist of the idea is that if you really want to conserve insects, you actually go to habitats which are stable, because insects have been living in those habitats for so many years. They have not been able to adapt because they have been exposed to constant environment, and so their genetic makeup has remained unchanged. And now because of climate change, the sun gets hotter over here. These organisms do not have any alleles which have been selected for tolerating hot environments. So when you talk about conservation, this is the conservation you should do. You should find pockets where you have a lot of stable climatic conditions. Go and look for the insects. In those insects, look at populations and check out the genomes of these and check if you can do anything. So if the genome is quite small, it has less number of transposable elements, there is a high chance that there is a bottleneck, and that is the population you should start focusing your attention for conservation. Okay, so now as we saw, so this is kind of a gist of what was discussed yesterday by Dr. Ramamurthy, that you definitely have a taxonomic scale to look after, but then you also have a lot of broader aspects. So earlier, when we really did not have a lot of features of identifying insects, we were just classifying a lot of things as butterflies. But now after we know that, okay, I know a butterfly, it has 5 families. Among that 5 families, I have 5 species. And in one species, in Himalayas, you have a darker moth. In South India, you have a lighter moth. You have a lot of knowledge about populations. So it is these populations which should be the focus of attention, because populations can be subject to extinction. A species doesn't go to extinction all of a sudden. A species might have a lot of geographical variant counterparts in different parts of the world. But if individual populations of that species go extinct, that's a sign that the species is not able to adapt. And to do that, maybe you should sequence as many genomes, which is why we have people like Saroja and their great companies to do sequencing at a cheaper costs. And that really is a powerful genomics, which we can employ together in conservation. So I think with this, we have combined ecology, evolution, physiology, genomics, and that is what I think was the gist of what you were talking, Dr. Ramamurthy. And with that, I think I'll end my talk and happy to take questions. So yeah, this is the review, and I leave you with this idea whether all of this is evolution or plasticity. Give a thought. Is there a choice?